Okay, so we have started recording. It's good to have everybody here for our monthly Cannabis Research Coalition Café, which meets on the fourth Wednesday of each month. We have been discussing propagation practices for the last several monthly meetings. And so this is our best practices series, and we will move on to other topics in the coming months. But today we'll be talking about nutrition in propagation, and then we'll provide some a recap of the last several meetings. And so today the speakers will be myself and Alex Carver. I will show some data that we have had from the past work that we've done on nutrition and propagation of some ornamental species, which I think provides some nice background information that tells a very similar story to cannabis. So we'll do a little background with ornamentals to start out with. And one of the concepts that is important is that we're trying to maintain nutrition in the cuttings that we're harvesting from the mother plants. And so when we harvest a cutting, it has so many nutrients in it. And what typically happens is demonstrated here. What happens to those nutrients, or that nutrient concentration, over the first week and a half in propagation. And so what you see here, this happens to be data with an ornamental species called Dioecia. We've done this with Poinsettia and some other species. They all kind of look the same. So we'll just show you this one today. So these are nutrient analyses done on the day the cutting is harvested. And what we have is phosphorus, which is the black, calcium, which is the yellow, and magnesium, which is the gray, on this axis. So this is the percent that of each of those nutrients that's in the tissue. On the right-hand axis, it's on a little bit different scale. And so this is potassium, and potassium is the blue. And so these are the initial concentrations. So we have, for example, 1.2% of the tissue of the whole cutting. This isn't just the leaves. You take that whole cutting and dry it, grind it, send it to the lab, and it's 1.2% calcium. And then what happens over 11 days in propagation is those concentrations become diluted. And so for example, with calcium, after 11 days, we are down to 0.7% calcium. And so we have large changes, almost 50% reduction in most of these nutrients over a week and a half. And this is assuming we are not supplying any nutrients during those 11 days. Keep in mind, these are just starting to root at this time, 11 days. So so a lot of growers would traditionally really not start fertilization until that initial rooting process. But this data suggests that if you are waiting till roots are initiating before you start fertilization, the plants have really limited nutrients available. What we think is happening primarily is that you're having a dilution effect, and that you're getting some growth on that cutting, but you're not getting much nutrient uptake. And so the nutrients that are there become diluted. So this is the story, and this is what we're trying to avoid. We're trying to avoid this big drop, and the way we avoid it is by supplying better nutrition during the first 11 days in propagation. Also, we can improve the situation by fertilizing the stock plant well enough so that the tissue numbers of the original cutting are nice and high. So if we think about it, there really are 4 ways that we can supply nutrients to cuttings. First would be through the stock plant fertigation program. So we fertilize the stock plants, nutrients go into the cuttings, and we kind of load the cutting with nutrients to help it get through those first couple weeks in propagation until they have a root system that effectively and efficiently takes up plant nutrients. The second means of providing nutrition would be through the mist system, and that would be putting a water-soluble fertilizer in the mist that is being sprayed on the cuttings in propagation. This is typically done at concentrations of about 50 to 75 parts per million nitrogen. We do have to be careful with phosphorus on some species and I don't know about cannabis, but for the best example is poinsettia. If we mist poinsettias with phosphorus, we get some severe leaf distortion. So we do have to be a little bit careful with mist, but most species are fine as long as the concentration of the nutrients isn't real high. So then the third option would be to supply nutrition through a starter charge in the propagation medium. So if you buy a commercially blended medium, often this comes with a starter charge. Often the products that are mixed into the starter charge are potassium nitrate and calcium, nitrogen, potassium, and calcium. Other things can be put in there too, but those are typical. You could easily test this by sending your media to a soil testing lab. You can measure the EC of the media, will give you an indication that there's starter charge in there. You take media such as Oasis foam or rock wool cubes tend to not have starter charges put in them. And some propagation media do not either. So you want to know what's in there. And then the fourth way of getting nutrition to cuttings would be through applying a nutrient solution during the fertigation process. Even though in propagation, we're not often drenching the media with much water because the mist is adding moisture or the dome is preventing a lot of evaporation. This is still a way that we can get nutrition into the cuttings. Just like when you harvest or take cut flowers and put them in a vase, cut flowers take up solution. So unrooted cuttings will take up solution even though they don't have roots. They just don't do it all that efficiently, but they do take some up. So the fertigation solution is the fourth option. A couple notes are that nutrients are lost in propagation when we mist, especially when we mist excessively and cause leaching. So if we put a pan underneath a tray of cuttings and that pan is accumulating water or solution or leachate, there are nutrients going to be in that solution. So we are losing nutrients when we leach. So this is the benefit of domes, is that we really don't waste any nutrients. And this is the problem with excessive misting. Now you can mist, and if you don't mist excessively, you don't get leaching. But this is something to be very careful about. And I'll demonstrate why in a few moments. The second note is that the faster a cutting roots, the faster nutrients can be taken up efficiently. And so you have better nutrient uptake through the roots, through the cut stem. So we tend to have fewer nutritional problems on cuttings that root fast. We tend to start to see nutritional problems if our cuttings are not rooting, starting to root by days 7 to 10. And so this is how actually rooting hormone can actually benefit nutrition. Sometimes you'll see nutritional problems in cuttings, and the problem is actually solved with rooting hormone because we're getting roots faster, so the nutrients that are there are getting taken up more quickly. Whereas if the cutting just lingers without roots, you tend to get more and more nutrient problems, deficiencies. So we'll go to an example that we did on petunia several years ago. This is a variety that all the growers I was visiting were having problems with. It happened to be just a particularly difficult variety. And in cannabis, I think we have quite a number of these varieties that are not that easy to grow, but they're good varieties, good strains that that there's a good market for. So we grow them even though they may not be that good of a plant for growing and propagating. And that was the situation here. It was just a bit of an oddball, because petunias in general are pretty easy, and this was a hard one, but it was a good guinea pig for experimental purposes. So what we did in an experiment, we looked at basically 3 ways that we might manipulate nutrition. So we stuck cuttings in a peat-based media without a starter charge. So there's no fertilizer to start out with. And then we had 8 total treatments, combinations. We had mist versus no mist. So instead of having a dome, we just put plastic, and underneath the plastic, we had some remade row cover fabric over the cuttings to help keep it, humid. And then we would actually hand mist underneath. We lift the plastic up and mist underneath every day just to keep it humid. And so we had mist, no mist. We had rooting hormone, no rooting hormone, and our rooting hormone of choice was a spray on day 1 of propagation with 500 parts per million of the potassium salt form of IBA. And then we had a no fertigation treatment and fertigation. And fertigation implied that we were drenching the media with 100 parts per million nitrogen using 15-5-15 every other day. So this is quite high, but we wanted to do something that was like the most you would possibly think of doing at that time anyway. So we did every other day with 100 parts per million. Okay, and then this is a picture of our 8 treatments. So on the far right are fertigation. The left-hand column is no fertigation. The top half, these 4 are no mist, meaning they're under plastic. Bottom 4 are mist. And then within each of those, we have no rooting hormone for these 2, rooting hormone for these 2, no rooting hormone, rooting hormone. And you can see there's some treatments up in this corner, like these 3 are particularly good. A lot of growth. These pictures are taken, I believe it was at 11 or day 14 after sticking. And you see there's some in here with, you can see iron deficiency. Here you start to see iron deficiency and phosphorus deficiency, the purpling. So you get a shoot tip yellow, like this cutting right here, that's iron. You get a shoot tip yellow and purple, that's iron and phosphorus. So we'll look at the 2 best, the best treatment and the worst treatment, which are the extremes. And so the best treatment was when we did no mist. We had the cuttings under plastic, so we had no leaching. We fertigated every other day with 100 parts per million, and we used the rooting hormone, KIBA. And so we pull these plugs out, show them up here, and you see this is again day 14, and every cutting you could pull the root ball out, and it held together very nicely. And so that was 100% rooting in 14 days, which for this variety was pretty remarkable because people were having real problems with it. And then the worst treatment, you can see here, only 1 of the 20 some cuttings that are here rooted. The rest just have nothing at all, or a really dinky root that didn't hold the root ball together. And you can see we have this iron and iron here and phosphorus deficiency. So, and this treatment was the opposite of the previous treatment. This had mist, this had no fertigation, and it had no rooting hormone. So these rooted very slowly and poorly, and hence you start to see nutritional problems popping up. So our conclusions were that we want to minimize leaching of nutrients out of the propagation media, and domes are helpful, but they're not required if the mist is properly managed, as long as we don't mist excessively. Fertigation before root initiation is very important. So we either have to have a starter charge in the media, which helps, and or add enough fertilizer through the mist, or we have a drench type of application, a fertigation application. So there's 3 ways we can get nutrients, and we may do some combination of those 3, but we need nutrition during that first week. That's the bottom line. And then rooting hormone is certainly very beneficial because, again, you pop out roots a bit faster and you don't see the plants linger with these nutritional deficiencies. So that's the ornamentals story, which I think will now show you how cannabis has some real similarities. And so I'll turn this over to Alex, and she'll tell you about she's run this experiment once. We have the second rep of it in the greenhouse now. And so this is preliminary, and we will update it in the coming months. But the first set of data looks good. And so we'll show you it today, but we'll have more conclusive data in a few months. So Alex.
Alrighty, thank you, Dr. Faust. So on this portion of the experiments for cannabis propagation, we're taking a look at how stock plant nutrition and nutrition provided in propagation affect the rooting success of, we worked with 2 cultivars for this experiment, Sunset and Jack, Sunset being the variety that is known for producing a large biomass, and then Jack is one of the varieties that produce good quality smokable flower with a good terpene content and dense buds. So these 2 cultivars were transplanted into our 2-gallon containers and grown with 200 parts per million nitrogen of that 15-5-15 fertilizer for 4 weeks. On week 5 is when I began the mother plant treatments of 100, 200, or 300 parts per million of the nitrogen. So these stock plants were receiving about 2 to 3 liters of nutrient solution a day. And it's important to note that we gave the mother plants these fertigation treatments for 2 weeks before harvesting cuttings from the mother plants. When we went ahead and harvested the cuttings, I set aside some cuttings to dry to send to a lab for nutrient analysis. And then the rest of the cuttings are planted in 3 different propagation treatments. So they are planted in a peat light media that has no starter charge. So we then drench those propagation, those trays with either 0, 100, or 200 parts per million of the nitrogen on day 1, and again on day 7 in propagation. We maintained these cuttings under domes to prevent any leaching. And then on day 14 is when we went ahead and we did our rooting analysis to take the fresh shoot weights and the fresh root weights. And so this is a little visual of our 9 different treatment combinations. So you have your 3 different stock plant or mother plant propagation treatments. And then those cuttings are then assigned into 3 propagation treatments from 0, 100, and 200 parts per million. And again, these were applied as a drench on day 1 and on day 7 in propagation. And so our results, we saw that propagation fertilization had a very significant effect on rooting or on the root weight of these cuttings. So if you go ahead and you look at the bottom, the x-axis, that is your stock plant fertigation treatments. And then your green line is what we saw was our best treatment, was your 200 parts per million treatment in propagation, combined with your 100 parts per million of the stock plant nutrition. And so it's important to see here that your propagation treatment is very significant or significantly affects your root weight. You'll see that our worst treatment was actually your lowest nutritional treatment, being your 0 parts per million provided at propagation as a drench, and your 100 parts per million, your lowest stock plant treatment. So that was our worst. So when there's low nutrient levels, you're not going to get a lot of rooting in propagation. But then as you see, as your stock plant fertigation increased, so when you're looking at your 300 parts per million stock plant treatment, that actually having nutrition in propagation decrease. There seems to be that there was nutrition in excess. And that's why you see that green line start to decrease as we increase the stock plant fertigation program. But also, if you have a stock plant that has good nutrition, so at your 200 and 300, then they performed better than your lower 100 parts per million of the stock plant treatment if there's no nutrition in propagation. And so you'll see that stock plant fertilization is important if there's no nutrition provided in propagation. But when you are providing nutrients in propagation, then your best treatments are going to be your 100 to 200 parts per million of your stock plant fertilization program. And so taking a look at the shoot weight, what you'll see is that actually with our increase of nutrition or increase of nutrients provided, you will also see the growth of the shoot or the shoot weight will increase. So when providing your 300 parts per million, your highest nutrition in stock and your highest nutrition in prop, you're going to get the greatest amount of shoot growth in propagation, because at the beginning, all of these cuttings are very close in weight. And so at the end of the 14 days, what you'll see is that based on the nutrition provided in stock and in prop, the more nutrition provided, the greater the shoot weight, which is not always ideal, because what we're looking for really is that root weight. And then in regards to our rooting percentages, it's important to remember that even if a cutting has a small single root, that is considered rooted in our experiments. But as you will see, that with excess nutrition provided, our rooting percentages actually decrease. There was a point where we're providing too much, or it seems nutrients in excess, and it's actually detrimental to our rooting and propagation. But again, looking at our 100 to 200 stock fertigation program and our 100 to 200 propagation program, you're going to see your best rooting percentages in that area. And so for our conclusions, our best stock fertilization program is going to be our 100 to 200 parts per million of the nitrogen. But it's also important that we are providing nutrition in propagation. And so our best treatment was the 200 parts per million nitrogen on day 1. So when you're planting the cuttings, and then again halfway through propagation on day 7. Our 300 parts per million stock plant treatment produced a larger shoot weight, but it also produced our lowest root weights and our lowest rooting percentages at the end of propagation. And so to kind of summarize all of the experiments that I have presented thus far, we're looking at recommending our stock plant nutrition to be anywhere from 100 to 200 parts per million nitrogen. Our cuttings are only about 2 to 3 inches, so it's not those tall 6 to 8 inch cuttings. We're using much smaller cuttings in propagation. We're recommending that the propagation media be maintained between 90 and 100% container capacity. Again, these cuttings really like to stay wet. The drier medias do not perform well in propagation. In regards to rooting hormone, that seems to be very cultivar or strain specific, but generally across all of the cultivars that we have tested, 2,000 to 4,000 of your potassium form of NAA or IBA as a stem dip performs well. And then in regards to nutrition in propagation, we're looking at a drench of 200 parts per million of nitrogen on day 1 and day 7, but that's also assuming that there is no starter charge in your media. And to avoid leaching nutrients in propagation, mist or dome works well, but you must have a good mist program to prevent the leaching of those nutrients. In our nutritional experiments, we use domes to prevent this, but in facilities or in commercially run facilities, you have to have a good mist system to avoid any leaching of these nutrients, and then not being able to have it provided to the cutting. And so if there are any questions, I know this is pre-recorded this time, you can go ahead and shoot us an email and we will get back to you as soon as possible.
All right, thanks, Alex. We hope you have enjoyed this portion of the CRC. I think the guidelines look pretty good. I think you can see that we're pretty regularly hitting up near 95% rooting after 14 days on most varieties when we're using the best practices. I think there's a lot of room for improvement. That what I see in the industry is often a lot below that number. So I think we can get our numbers up. If you have questions you like or want to have a discussion about your particular system and how we can get rooting up in propagation, we'd be glad to entertain those questions and discuss your specifics if we can be of help. So for Alex, Allison, and myself, thank you for attending, and we'll see you again next month.